Hey there, biology students. Happy Friday. So I'm going to take you through the process of protein synthesis, which is the process of going from DNA inside of your cells to the proteins that make up your bodies and minds. So where do we start? We start within a living cell. We are deep in the center in a structure called the nucleus. Within the nucleus, there are tightly wound bundles. These are structures called chromosomes. If we unwind them, we'll find a long molecule of DNA. DNA contains short stretches of nucleic acids that we know how to read, right? These are short stretches that code for certain traits like eye color or right or left-handedness. Right? These are called genes. It's what you pass down to your offspring, what you inherit from your parents and grandparents. So this is a chart from your homework slide. Protein synthesis occurs in two phases. First is the conversion of DNA to mRNA. This is called transcription. The second phase is the conversion of mRNA codons to an amino acid sequence. This is called translation. So first, let's go over transcription. This is inside of the nucleus, remember, and the brown little blobby thing you see here is RNA polymerase. This is where this reaction is happening. Okay, so the DNA molecule, the double helix, gets split into two pieces, and that leaves open an opportunity for these little RNA nucleotides to come in and form a messenger RNA molecule. Okay, and that messenger RNA molecule is going to swim. It's going to be released first from the RNA polymerase and then swim outside of the nucleus. All right. So how does that mRNA molecule know how to form? Well, it follows a very specific set of rules. Every time DNA has an adenine, mRNA matches with a uracil, okay, every single time. All right, so then you form these three-letter words called codons. All right, A is converted to U, C to G, C to G. Okay, and then in pRNA or transfer RNA, which we're about to talk about, you have anticodons. Anticodons are made following the same rules. A, U to A, G to C, G to C. Right, these are the tRNA anticodons. Every tRNA molecule has 3 bases on it, and it matches up with 3 bases on messenger RNA. This is the transfer RNA. tRNA molecules are also attached to amino acids, which are the building blocks of proteins. So how do we know which mRNA codons translate to which amino acid sequence? Well, one codon translates to one amino acid. And the only way we can find out which is by looking at it on page or slide 3 of problem set number 2. You can see our 3 codons up here are tryptophan, proline, and isoleucine. Okay, so look at the right side of your screen. You have a tRNA molecule attached to an amino acid coming to join the ribosome. And on the left side of your screen, you have a tRNA molecule without an amino acid. And at the top half of your screen, you have a growing chain of amino acids. So what happens? The tRNA molecules bring a unique amino acid to add to a growing amino acid chain. Right? Once the end of the mRNA molecule is reached, the amino acid chain folds into a protein, and that is protein synthesis. Okay, so the last part of your homework is to translate an amino acid sequence or protein into a trait. So this chart is also on slide 3 of problem set 2. And you can see tryptophan, proline, and isoleucine translates to a green body that walks on 2 legs. And that is protein synthesis. Happy Friday, everybody. See you next week.